some of these other projects and emails so I'm not distracted, and we will get started. Okay, so welcome everyone who is participating in today's Zoom extension meeting organized by Ashley Thompson. Today's speaker is Kelsey Galimba. Kelsey is the new assistant professor of horticulture here at the Mid-Columbia Agricultural Research and Extension Center. Kelsey received her BS in plant pathology from University of California, Davis, and her PhD in biology from University of Washington, where she characterized genes responsible for floral organ development. She then completed her postdoc with USDA ARS, using molecular and horticultural approaches to study fruit development in apple, peach, and plum. She joined McCarrick as assistant professor here in November of 2020 and is currently developing projects and plans to address issues related to fruit quality and yield in pear and sweet cherry. Overall goal of her research program is to optimize orchard production systems for these two crops in order to enhance the efficiencies and competitiveness of our Mid-Columbia growers. So if I'm co-hosting with Ashley today, so if there are questions, put them in the Q&A part. I see there's some chat starting in the chat, but if you have specific questions, I'm going to try to filter those and help Kelsey out with those, so just put those in the Q&A. Other comments can go in the chat, and with that, I will let Kelsey take it away. Kelsey?
So I'm seeing the host disabled participant screen sharing.
Oh yeah, I think Ashley needs to make you a host so that you can share if she's still with us. I don't have those controls here, I don't think.
Oh, here, it's working now.
She's got you.
Yep.
And I see your screen. I'm going to go ahead and mute.
Great. All right. Thanks. Thank you, Chris, for the introduction. And thank you, Ashley, for inviting me to give this webinar. Thanks also to everyone who's attending. So like Chris said, I've been here for about 4 months now. I started in November. Unfortunately, I don't have any new data to share with anyone. So Ashley and I thought that this would be a really good opportunity for me to kind of give an overview of my background. And then I'd also like to talk about the work that I'm going to be starting up my first year here this next spring and summer. So as an overview, I'm going to talk a little bit about the work I did in my PhD. That was at the University of Washington, where I worked on flower development. I'll talk in quite a bit more detail about my postdoc research. That, again, was at the USDA ARS Appalachian Fruit Research Station in West Virginia. And there I worked on tree fruit development in apple, peach, and plum. And that's, I'm going to talk more about that because it's quite a bit more relevant to my current position, which, like Chris said, is an assistant professor of horticulture. I study pear and sweet cherry here at MCARRIC in Hood River, Oregon. So I'll talk today just really briefly about what I have plans for this summer. So there are a few ongoing trials here at MCARRIC, Todd Einhorn's quince rootstock trial and Pear McCord's cherry breeding trial that I'll be participating in. I'll talk about some PGR trials that I have planned. I'll go a little bit into my involvement in the cherry cold hardiness model project, and then also touch briefly on a new project that I would like to start looking at improving fruit set in pear. So to start with my PhD, the premise behind my PhD, so I studied floral development, and the premise behind this field of research is that we know that changes in floral morphology, so changes in the way that flowers are shaped, their color, their smell, all of these different characteristics, we know that that really drives diversification. And we know that because flowering plants right now are currently by far the largest group. There are way more flowering plants than there are non-flowering plants. So we know that, differences in flower morphology are really important for evolution, but up until recently, we really have not understood how genetics are involved in that. So what are the genetics behind how different floral forms arise? The lab that I was in for my PhD was the Distillio lab at the University of Washington, and we focused on a set of genes called the Floral Organ Identity Genes. And like their name, these genes are necessary for the development of all of the different floral organs. The way that they were first characterized was by using model organisms or model species. When you're in flower development, the model species are Arabidopsis, Snapdragon, and Petunia, actually. So all of these genes were kind of characterized in those model species, and they were all put together into this nifty model called the ABC or the ABCDE model. And the way that this model works is that all of the different floral organ identity genes are assigned to different classes, and then different combinations of the classes are needed in order to get development of each organ type. So for example, in World 2, if you want to get petal development, you need a combination of B, A, and E class genes. The way that this model was put together was actually by knocking genes out. So that's shown in the diagram on the right here. And this is actually in Petunia. And you can see when you lose function of one of these classes of genes, you get really interesting phenotypes. You get really dramatic mutations. So we call these homeotic mutations, where the identity of a floral organ is actually transformed into a different identity. I'll point out the C mutant. So a lot of people are actually familiar with C mutants because we also know these as double flowers. And most of our cut flowers or garden flowers have double flower varieties available. In our lab, we used a species called Thalictrum phthalictroides, or rue anemone. And Thalictrum is a really interesting species because it actually diverged earlier than most of the model organisms that have been studied. Thalictrum has a lot of kind of basal floral characteristics. So we were interested in seeing how these genes differ in a kind of a more basal species. I'm not going to go into the details of what I studied in my PhD. There's, I did a lot of gene characterization. So there's a lot of kind of, very specific genetics, but I kind of wanted to give an overview. So I took two major approaches during my PhD. One is kind of a forward genetics approach, and forward genetics is where you start with a phenotype. So a lot of times this is a naturally occurring mutant, and then you figure out the genotype behind that. So you take a look at what genes are causing that phenotype that you're seeing. The wild type Thalictrum phthalictroides is in the middle there, and then these are all horticulturally available mutants that we characterized. So we characterized either mutations in these different mutants or different cultivars, or we characterized gene expression shifts. And so, these mutants, they occurred spontaneously, either in the wild or in somebody's garden, and people propagated them, and you can now buy them commercially. The bottom three are double flower mutants, and so we showed mutations that were responsible for these phenotypes. We also showed some gene expression shifts that were responsible for the top two. So the Betty Blake at the top there has no male reproductive organs, and then the green hurricane in the middle, which is also called green dragon, that has a lot of these homeotic mutations where the stamens are actually partially transformed to other types of perianth organs. I also took a reverse genetics approach. So reverse genetics is the opposite, where instead of starting with a phenotype and figuring out the genetics, you're actually manipulating the genetics and then observing the resulting phenotype. So in my case, I was knocking out genes. I showed that when we knock out genes in the C class, we get this double flower phenotype that you see on top. I also showed that by knocking out genes in the B class, I could disrupt both stamen development and also the showiness of the sepals. So in Thalictrum phthalictroides, there are no petals, but they have petal-like sepals. I also did some work with ovules, so the structure that becomes the seed. And here you can see at the top left, there's a wild type or a normal ovule. And if we disrupted or downregulated another gene in the C class, we got abnormal ovules that would actually abort and not set seed. So I did some work in ovule development as well. So that's all that I'll say about my dissertation. If you have questions, definitely let me know. But it's a little bit kind of more further related than to what I do now than my PhD or than my postdoc work is. So I'll switch gears now and talk about my postdoc. When I took my postdoc position, it was a really nice transition because I went from working on floral development to working on fruit development. And as it turns out, a lot of these floral organ identity genes that are so important in flowers also have really important roles in fruit development later on. So that was really nice. I was also really excited because we know that fruit morphologies are just as diverse as floral morphologies, but we actually know even less about how fruits develop or how fruit flesh develops. This was also kind of a really exciting switch for me because I think I was more interested in trying to work on things that had more of a real world application. So working on fruits, of course, is important because fruits make up a large part of our diets. So this is showing a bunch of different fruit types in the rose family. These are all really closely related species within the Rosaceae or rose family, and there's a bunch of diversity here. When I first started, I was on a project that was funded by the NSF, so it was kind of a basic science project. And here we were kind of trying to do a comparative analysis of early fruit development within the rose family. So we specifically wanted to know which genes were initiating and directing fleshy fruit specification from different floral parts in 4 species from this rose family. So I was working on apples and peaches, and then we were collaborating with the University of Maryland, who was working on raspberries and strawberries. All 4 of these species their fruits look very different, but they're actually closely related. They're also all derived from an ancestral dry fruit. So they have become fleshy, but they have become fleshy in different ways. The apple, we know that is an accessory fruit. An accessory fruit is where, the fruit flesh, the part of the fruit that you are eating is actually not coming from the ovary. In the apple's case, and this is the same as pears, all of that fruit flesh is coming from a hypanthium, which is kind of a cup-like structure that surrounds the ovary. The ovary itself forms the core of the apple or the pear. Peach is a drupe like cherries, and that is a botanically true fruit where all of the different layers of the peach are derived from ovary. The raspberry is a drupetum or a collection of small drupes, and that's because there are multiple ovaries in a raspberry flower. A strawberry flower also has multiple ovaries, but those ovaries turn into achenes or dry fruits. And the red fleshy part of the strawberry that you eat is actually derived from the tip of the stem or the receptacle. So this was a large project and we took a lot of different approaches. We did a lot of work describing the morphology of both flower and fruit development in these 4 species. We did comparisons of gene expression before and after pollination and fertilization in all of the different floral parts. We did some investigation of gene function, and so that was by knocking out genes. Most of that was actually done in strawberry, but I'll talk about one specific example where I did some in apple. And we also investigated fertilization-induced hormone production. And so that was an area where I got interested in, and I got interested in PGRs as kind of an offshoot of that portion of this project. So I don't want to talk too much about the gene expression analysis that we did. I will show one really interesting gene that I was particularly focused on, and that's the gene PISTILLATA. PISTILLATA is interesting because it has been hypothesized to be a suppressor of flesh development. So when it's expressed, it keeps fruit from developing. And we saw gene expression that kind of supported this hypothesis. So on the left-hand graph, we have PISTILLATA expression at different developmental time points in apple, and there are 3 different tissue types. The yellow hypanthium, you can see that the expression is high at 0 days, so that's when the flower opens. And then by 5 days after anthesis, when pollination has occurred, we get a large drop in pistillata expression. And so that indicates to us that pollination is somehow triggering the downregulation of pistillata, and then that's what allows the hypanthium of the apple to develop. In peach, we saw a different expression pattern. So peach had higher expression overall, but that just kind of gradually dropped in the hypanthium. And that's probably because the hypanthium of the peach, the cup, it doesn't become fleshy at all. It just kind of dries up and dehisces, falls off as a shuck. So I was also interested in pistillata because there are a number of cultivars in apple that are naturally parthenocarpic, and they've been shown to be pistillata mutants. So parthenocarpi is not something that happens very frequently in apple. It happens more frequently in pear. But in apple, there are these two cultivars, Spencer seedless and Wellington bloomless, and they will set fruit parthenocarpically. They do not need to be pollinated. But they do have strange looking fruit, and that's because they're pistillata mutants. Pistillata is actually one of these floral organ identity genes. So it's needed in order to get petal and stamen development. When you don't have pistillata, you don't have petals and you don't have stamens, and you have a lot of extra carpels. So what's happening with these fruit is these extra carpels are kind of forming a strange phenotype in the mature fruit. You can see in the Spencer seedless picture, there are a lot of kind of extra papery structures at the blossom end. There are some extra lobes a lot of times, like you see in the Wellington bloomless. So this is kind of an unfortunate phenotype. And we also know that both Spencer seedless and Wellington bloomless are both very poor quality eating apples. They don't taste very good. So I was really interested in figuring out if we could manipulate pistillata expression to try to, create parthenocarpic apples that don't have this multicarpal phenotype. And so before I left AFRS, I made a number of constructs. We were using a lot of our other gene expression data to try to do this in a really fine-tuned way. So knocking down pistillata, but very specific time and very specific um, regions of the flower so that we don't get this multicarpal phenotype. And that's being transformed into gala apples right now. So it will still be a number of years until we know whether or not this worked. But this is something that I thought was kind of a cool project. I'll start talking now about some of the work that I did on PGRs. So again, this was kind of stemming from that NSF project where we were interested in plant hormones in relationship to pollination and fertilization. There are a number of PGRs that are registered for use in apple to do things like increase set or improve shape. Perlan and Rio are registered to increase fruit set and also improve size and shape. Prolin is a PGR that is registered to actually rescue yield of frost damaged flowers. And frost damage is something I had a lot of experience with my first year as a postdoc. We had a really late frost in the spring and we lost everything. So everything that I was supposed to collect that year was frozen and died. And that was kind of my welcoming into the tree fruit world. So the idea behind this project is that these PGRs have these great uses, but there's also a lot of reported negative side effects. So most of those side effects kind of go along with increased or advanced ripening. Apples that have been treated with the hormones in these products sometimes are softer, they're less acidic, they have higher sugar, less starch, a lot of indications that they are ripening too quickly and that they probably wouldn't hold up in any sort of cold storage. So we wanted to look into that in a little bit more detail. I started out by testing a number of different hormones. So we didn't use any of the commercially available products. We just used the straight hormones. So I used gibberellin or gibberellic acid, GA3. I used a synthetic auxin, NAA. I combined the two of those. I also used NPA, which blocks the transport of auxin. And then I had a negative control and two pollinated controls, hand pollinated and open pollinated. And on we measured fruit retention and also fruit size after applying these chemicals once. And we found some really interesting results with the GA3. So if you look at the last data point in the fruit retention graph at harvest, we had a number of fruit harvested from the hand pollinated and the open pollinated controls. And we had also a significant amount of fruit harvested from the GA3 treated control. Not as many as the pollinated controls, about half as many. All of the other treatments, we had no fruit left at harvest. So we really focused in on that GA3. Taking a look at fruit size throughout development, the GA3 treated fruit also had similar area to the pollinated controls. So these fruit were large. They were just as large as the pollinated controls. The other two combinations of hormones, so the NAA and the GA3 plus NAA, we saw some fruit retention, at least up through June drop for those hormones, but the fruit stayed incredibly small, so they never really grew. They stayed on the tree, but they were completely stunted. So GA3 is what we really followed up on because that we got the most interesting result from that hormone. We showed that this was parthenocarpic development, so we had excluded pollinators, and then we quantified the number of seeds that we saw at harvest. GA3 treated apples were seedless. They had no seeds. We also saw this interesting difference in size. And so this is also, or not size, shape. This is also something that has been reported. GA3 treated fruit are longer, they look longer, and they look thinner, more similar to a red delicious kind of phenotype than the normal Honeycrisp phenotype. These are Honeycrisp apples. So we were interested in that shape difference. And we quantified it a little bit in a little bit more detail. So we showed that GA3 treated apples are thinner because their cores are thinner. If you take a look at the fruit length didn't have any differences, but the GA3 treated apples were statistically thinner than the pollinated controls, and that was because the ovaries or the cores are actually thinner. So we didn't see any difference in the hypanthium width. You know, that portion of the apple that you eat is still just as wide, just as large, but the cores are quite a bit thinner. And so we were interested in following up on the reason for that. We didn't see many of the differences that have been reported in the literature. So we had no difference in firmness, no difference in starch content, no difference in sugars. We did see a difference in pH. It was slight, but it was statistically significant. So the GA3 treated fruit were less acidic. They had a higher pH. And so we were interested in trying to take it a step further and figure out if we could discover what genes are involved in those quality differences that we're seeing. So we have the shape difference and we have this acidity difference. To look at the shape difference, we looked at the genes that were expressed in the core, in the ovary. Some of the gene pathways that were upregulated in that wider hand pollinated ovary, one in particular was interesting, and that's cell division. We thought that these cell division genes are likely, involved in this width, the size difference that we're seeing. We also looked at pathways that were upregulated in the GA3 treated ovary. And so here we were really interested in this hormone metabolism because we thought that hormones are very likely playing a role in the size difference that we are seeing. I'll show just kind of briefly some of the interesting genes that we pulled out of this analysis. So this is an example of RNA-seq data. This is a heat map. So we have 3 replicates of hand-pollinated ovaries, and then we have 3 replicates of GA3 treated ovaries. And where the boxes are red, that's where that gene is expressed highly. Where it's blue, it's expressed low. And so, for example, we have this gene that's associated with apple fruit cell division, and that's been shown in other studies to have that role, and it's expressed low in the thinner GA3 treated ovary. This cyclin gene that has been shown to inhibit apple fruit cell division, that's high in the thin GA3 treated ovary. We also took a look at the hormone metabolism genes. These all ended up being genes involved in cytokinin. And for example, we've got this degradation, cytokinin degradation gene that's expressed high in the GA3 treated ovary. We also found a gene that's involved in the sequestration of cytokinin that's expressed high in the GA3 treated ovary. So it's really looking like the difference that we're seeing in ovary width here is down to cell division, and that's probably being driven by differences in cytokinin levels. So, you know, this would need to be followed up with more. We didn't take this any further, but you know, I would suspect that if you have a cultivar where shape is important, where you don't want to see that red delicious phenotype, then PGR formulations that contain both GA and cytokinin would probably be preferable to just GA or GA3 by itself. We also looked at genes involved in acidity. So MA1 is a gene that recently has been shown to its expression levels correlate with acidity in different cultivars. So if you have high acid cultivars, you have high expression of MA1. And so we took a look at MA1 in our GA3 treated fruit, and we saw that at least early in development, not so much at harvest, but earlier, about 18 days after treatment, we have lower MA1 expression in our lower acid GA3 treated fruit. So it's really likely that for whatever reason, this MA1 gene is behaving differently in the GA3 treated fruit, and that's why we see a difference in pH. And we can, you know, take this kind of a step further and say that maybe if you have a cultivar where tartness is important, like say Granny Smith, maybe you wouldn't want to use GA3 in those cultivars. The difference that we saw was small. We didn't actually taste these fruit. I'm not sure if you would be able to taste the difference. You know, it was statistically significant. So maybe if there's a cultivar where, you know, acidity is more important, that would be something to keep in mind. Another project that I worked on also had to do with PGRs. And so this project was actually looking at PGR use in prune plum. The cultivar Improved French is the prune plum cultivar that's grown in California. This was a collaboration between us and the University of California, Davis. They have an issue with prune plum. They've been having this issue more and more recently, where they get high spring temperatures and that really decreases their yield. So the graphs here on the left, they only go through 2007, but this is something that has been continuing on since then. In years where their max temperature exceeds 27 degrees Celsius during bloom, they have very decreased yields. And so Franz Niederholzer, who's an extension agent with UCD ANR, reached out to us and asked if we could collaborate by using a system that we had at AFRS called our FasTrack plum system. And the FasTrack plums are this really cool system where they overexpress a flowering gene. They were created to actually accelerate breeding. They were used for that purpose. But what's neat about them is that they flower and they fruit very precociously. So almost immediately coming out of tissue culture or after being budded, they will start setting flowers and setting fruit. And they stay small, so you can grow them in the greenhouse. And so we thought this would be a really good way to, you know, manipulate temperature in the greenhouse and study whether or not the PGR Re Tain was having an effect on a fruit set. So there were two purposes. One was to try to gather data to support the use of Re Tain in plum, again, to try to increase yields when you have high spring temperatures. And then, you know, on our side, we really wanted to try to develop this FasTrack plum as a model system so that we could use it to study PGR applications year round. First we determined whether or not the response to Re Tain was similar between Improved French and FasTrack plums. You know, they're different genotypes, so we wanted to make sure that they were responding to the PGR in a similar way. And then we needed to increase the number of FasTrack plums in the greenhouse. These FasTrack plums We still had them, but they were not being used for breeding at that point, so we had very few of them, and we needed a lot in order to do the experiment. And then eventually, the plan was to evaluate the effect of retain on plum fruit set at both high and normal temperatures in the greenhouse. So this is something that I left before it was finished. Before I left, I collected data on whether or not retain had a similar effect on improved French and FasTrack plums in the field. And long story short is that it does. We saw a little bit of difference in the beginning, but the retention rates between both control and retain treatments in both FasTrack and improved French, they equalized by about 4 weeks after anthesis. So that reassured us that, these FasTrack plums would be a good model for testing PGRs for PGR use in improved French. And this project, before I left, was running into a lot of problems. So I had that second point there that was to try to increase the number of FasTrack plums. I didn't foresee this being a problem, but it actually turned out to be a really large problem. We had a lot of issues with incompatibility. We first tried to bud FasTrack plums onto Bailey, which is a peach rootstock. That didn't work. We tried Krimpsk. That didn't work. We ended up having success grafting onto Bluebird, which is actually a plum cultivar that was released from AFRS. It's a scion cultivar, but that seemed to work. We also had quite a bit of success actually rooting the FT themselves on their own roots. And right now I know that the FasTrack plums are in tissue culture. So that's also probably going to end up being a good way to clonally propagate these, but that takes a little bit longer. So by the time that I had left, we had a number of trees. They were in cold storage, and then recently they've started to do experiments with retain with them. And unfortunately, I don't have any data because this year was, I think we've still got to try to figure out how to optimize this as a model system. We got flowers, we got trees blooming, we got some fruit set, but the fruit set levels were very poor just all around. They didn't set well with the treatment or without the treatment. They didn't set well in the hot or the cold. So this is something that we're going to have to fine tune. And I think it's going to be, I think once we get it all figured out, it's going to end up being a really cool model system. So this is still ongoing over in West Virginia. And I hopefully that will produce some good data soon. I'm going to switch gears now and talk about the projects here that I'm going to be working on. So some of these projects I've kind of inherited, some of these projects are new, but these are the things that I'm planning on working on here in the near future. So one is Todd Einhorn's quince rootstock trial. So the goal here was to identify good, cold hardy, dwarfing quince rootstocks for the Pacific Northwest pear industry. And I think this project has been going on for about 12 years now. Todd started by identifying 20 quince accessions. He used the germplasm collection in Corvallis, and he found 20 that were identified as being equally or more cold hardy than Old Home by Farmingdale. They then took 9 of these accessions and they made 2 field trials. So one is here in Parkdale, Oregon, and then there's another up in Wenatchee, Washington, and Stefano Musaki is the person who is coordinating that trial. And they found that several of the accessions look really promising. They're precocious, they're size controlling, so they're dwarfing, and they're productive for Bartlett and Onjo. So this project we hope to continue on into the future. We would like to continue to assess the performance of the 9 established quince accessions here. I think these trees are coming into their 5th year, so this is a really great time to start seeing how these trees perform a little bit longer term. And then we also have plans to hopefully propagate the remaining 11 promising sessions, and that's with a collaboration with North American Plants. And so once those are successfully propagated, those will also be tested in a field trial. I'm also going to be managing the WSU Sweet Cherry Breeding Program Phase 2 regional trial that's here in Hood River. So this is a trial that belongs to Per McCord, who is the WSU Sweet Cherry breeder. And here we are going to continue to assess performance of 5 really promising sweet cherry selections that Per has here. And we are also going to each year incorporate additional selections that his breeding program are going to be advancing from stage 1 to stage 2. I'm also going to be doing a number of studies with plant growth regulators, so I'm continuing my work with PGRs. In 2021, I have 3 trials planned. One is working with New Farm on a PGR that has the goal of improving fruit set retention and size in cherry. I'll also be doing 2 trials for Valent, one on a PGR to improve fruit set and pear organically, and another that is going to be a post-bloom thinner in pear. So I'm excited to continue working on PGRs. I'm also getting involved in the cherry cold hardiness model. So this is another project that has been going on for a very long time. Started out here at MCARIC. So I think Todd Einhorn and Dave Jabot started this project. And there's been a lot of data that's been collected. And right now, we are trying to make a cold hardiness model from that data. So the purpose of the model is that it will use growing degree hours, and that in the future at any particular time during the spring, you should know based on the growing degree hours that your location has accumulated what hardiness level your cherries are at. And so there's been a bunch of data collected. When I started here, we were doing additional collections. So there were 4 additional collections that were scheduled to be made. 3 of them were in Prosser. We agreed to do 1 here at MCARIC in Hood River. And since then, the Oregon Sweet Cherry Commission has given me additional funding to increase the number of sampling, increase the number of collections that we do. So we've actually doubled the number of collections. We're getting a lot of data now from the Dalles area, so at 2 different locations in the Dalles, in addition to the one here at MCARIC. And we're hoping that that data is going to help build the model and help verify the data that we gathered in the past. So right now, every week, we're calculating LT10, LT25, and LT50. Those are the temperatures at which 10, 25, and 50% of the floral initials are expected to die. We calculate that every week, and that's being uploaded on the AgWeatherNet site. And there's also a beta mode for the cold hardiness model. And so again, it's a beta mode, and I think it's definitely going to be improved with the data that we're collecting this year. I'm also really interested in comparing the different methods that have been used to collect data in the past and also currently. So this is also funded by Oregon Sweet Cherry Commission. We have 2 identical freeze chambers here. So this seemed like a really good opportunity to me to compare the 2 different methods that have been used. One is DTA, or differential thermal analysis, and one is the kind of a more traditional freeze dissect method where you freeze buds and you pull them at different temperature increments, and then you cut them open to see how many are dead. And the 2 graphs here show the different types of data. So at the top, you're basically looking at voltage peaks and what temperature those voltage peaks occur at. That's a temperature where ice is forming inside the floral initial, and that tells you at what point they died. And then in the freeze cutting method, again, we pull a number of buds at each temperature, and we cut them open and we count how many are dead. And so then we can plot that and we can make estimates for LT10, LT25, and LT50. And so I'm really interested in, it's a nice opportunity to compare these 2 methods directly. I think that's going to be important for not only validating data that we're using that's been gathered in the past and currently, but also kind of informing us as to what method is really the best method for use in the future. If we end up collecting more data to either improve the model or verify the model or add more cultivars to the model. So that's something that I'm excited to do. I also recently got funding for a small project to try to use biomarkers to determine dormancy status in cherry buds. So the idea here is that there are a number of papers that have looked at gene expression changes throughout dormancy. So there are 3 stages of dormancy: paradormancy in the fall, then you enter endo or true dormancy, and then once your chill requirements are met, you enter ecodormancy, where the dormancy of the bud is being controlled by the temperature and not so much, initial internal control. And so there have been a number of genes that have been found that change. Their gene expression patterns change drastically depending on which dormancy phase you're in. And so my idea was to use published gene expression data to try to test some of these genes in Pacific Northwest cultivars to see if we can gather data about chill requirements and dormancy, and then add that into the cold hardiness model. Lastly, I'd like to talk about a new project that I'm excited to start. So I really want to work on improving fruit set in pear, specifically in Anjou. I've been told by a number of growers that fruit set in Anjou, you can have either initial set be low, the initial fruit set, or you can have excess drop that occurs. So you get a good looking fruit set, but then when June drop comes around, for whatever reason, you lose a large portion of fruit. This is a really interesting issue. I think it's going to be complicated because I think there are many potential factors that could be at play. Some of them have been researched more than others. I think it could be an issue with pollination. That could come down to bee activity. It could come down to the effective pollination period, so how long the stigma and ovule stays viable. I think it could be issues with fertilization. That would be like compatibility or S alleles. And I know that there's been quite a bit of work done on S alleles. Stephanie Musaki has done a lot of work on S alleles in pear. This could be something related to nutrient imbalances. It could be a carbohydrate deficiency. It could even be something related to the morphology of the pear. So maybe there's something going on with the pedicels and anjos that make them drop fruit more than other cultivars. So this is something that I'm interested in tackling. I think in this upcoming year, I'm going to focus on one aspect of that, and that's trying to improve set in high spring temperatures. So this is very similar to the work that I did on in plum. And I think it's kind of timely because I've been told that 2020 here had high spring temperatures that affected fruit set in Anjou. So this is the little graph here is just weather data pulled from our weather station here at MCARIC. Our full bloom for Anjou was April 7th. And we saw temperatures, close to 80 degrees in the days following that. So I think that that's very likely what's going on. For my research plans, so this is something I still need to figure out. So I need to be able to artificially manipulate temperature in the field. We don't have a greenhouse-based system with pear, so I need to be able to do this on trees that are in the field. I'd like to try to design and construct some sort of hot box structure so that I can heat up flowers or fruit. And then that hopefully will help me to determine the physiological cause of the poor fruit set because of the high temperatures. Because again, there are a lot of different things that could be going on. And then once we know why exactly high temperatures affect fruit set, we can then test the appropriate methods to try to increase set, whether that's a PGR like Re Tain or whether it's something else. So that's something I'm really interested in getting into this year. And that's just kind of that's the sampling. Those are the projects that I have planned. I have a large acknowledgement list here when you talk about your PhD and your postdoc and also your current work. There's a bunch of people you have to thank and a bunch of funding sources you have to thank. So I'd like to say thanks. And I can take questions now.
That was a great talk. I love the rainbow. That's how it feels every day out here.
There are so many rainbows here. I've
So
seen
many-
more rainbows since I moved here than I've seen in my entire life.
I know, yeah. I think the Irish are jealous of our
I
rainbows.
think they must be.
It's great here. I don't have any questions coming in yet, but I was really fascinated about this Android fruit set problem and all of the possible contributing factors. How do you begin to unpack and untangle all of those? Can you
Yeah.
really control? I mean, the hotbox idea is really interesting. Can you really control just one of those many, many variables, you think?
Yeah, I think that's all I can do. And I do think this is probably going to be kind of a multi-element issue. So it's probably not just one of those things that's causing fruit set issues. It's probably combinations of them. So it is going to be tricky to try to figure out. I think that the heat issue is something that's probably going to be easiest to separate from the other
Mhm.
issues. So that's why I'm focusing on that.
Okay,
But yeah, we'll see. We'll see.
that's a big project.
Yes.
I think that Steve was telling me that there was some crazy contraptions that were built in a previous
Yeah,
generation, and I think we have some of the remnants of those where
we
they
don't. I
drag.
asked.
Oh no. Oh, okay.
Yeah, they're gone.
Okay. Okay. I know there's an electrical outlet in the middle of one of those blocks that
Oh, that's good
was
to know.
I think it's disconnected now, but I think there was somebody building houses and running power and heaters and
Yeah, I'm gonna have to figure out the best way to do that. And unfortunately, there's not a lot of I mean, this is something that's done in different places. You know, people use baggies to increase temperature in one fruit. It goes all the way up to, big, massive whole forest enclosure type things.
yeah,
So
yeah.
I'm
There's
gonna
a
have to
couple
figure out.
questions that came in here.
Okay.
In S. Hanrahan, are you planning to look at pollinators,
Like insect pollinators?
insect pollinators?
Probably not, but maybe that would be something that Chris and I could collaborate
I would
on.
love to collaborate on that. If there's interest
Uh,
from the fruit growing community to explore the role of pollinators, Andoni Methanopoulos would be a great partner, and I'm sure we could figure out some way to collaborate with you and put in a proposal. Um,
yeah, that would be something I
Another
would be interested
question,
in.
follow-up question from an S. Are you planning on evaluating the Bell pair?
The Richard Bell's gem pair.
Uh,
Uh, I'm
I don't
assuming
know if
that's what
that's what I think that might be what she's referring to.
I'm assuming that's what you're referring to it as. Yeah, that's something I know Ashley is interested in as well. I would definitely be interested in that pair. I've heard a lot about that pair since coming here. Um, so yeah, if that's something that people would like to see, that would definitely be something that would be of interest to me. It seems like it's a great pair. I haven't tried it, but it seems like it's got a lot of good characteristics.
There's a couple more questions here. Mike Willett wants to know what, if any, collaboration might be done or planned with Matt Whiting's lab on cherry bud hardiness.
Yes, I need to talk to. So I just met Per McCord yesterday. I still need to talk to Matt Whiting. I know that they share a grad student who's working on a lot of similar things. So I definitely need to meet with him soon and try to figure out how we can collaborate on that, because I think that there's a lot of potential there for us sharing ideas and
Yeah,
experiments.
there's a lot of great collaboration opportunities out here. Everyone's been really welcoming for me.
Same here.
Gavin asked, how would heat lessen or minimize carb allocation for fruit set of pear?
Yeah, I'm not sure. I'm not sure about heat. I mean, when you think about carbohydrate deficiencies, you're thinking about solar radiation, cloudy days. So that might play a role. I'm going to have to dig a little bit more and see if there's a tie between temperature and carbohydrates, because I'm not clear what relationship there is between those two right now.
There's a lot to understand. We've got a very complicated job. That seems to be all the questions we have. I can give people another opportunity, but we're about 10 minutes away from 11. If people have other questions, I'm sure Kelsey would love to get an email from people
Yes, I would love emails.
and
Absolutely.
we're all restricted on the number of people we can physically interact with, so it's hard to go meet people, but I'm sure she would love to chat by email or Zoom and get to know people. I know her and I have both talked about the complications of meeting and greeting without killing anyone.
Yes, COVID makes
COVID.
everything hard. Yes.
Yes, yes. We have been told that we're not allowed to travel, so it makes it really hard to meet people.
Right.
But thanks everyone for participating and providing some questions and giving us an hour of your attention. I'm sure that we look forward to working with everyone here in the future.
Yes. Thank you so much.
Thanks for your time, Kelsey.
All right. Bye.
Thanks for putting everything together, Ashley. I know there's no pesticide credits on this one, but do you have anything else, Ashley, you want to wrap up with?
No, thank you, Chris, for stepping in for me today. I really appreciated it. And I look forward to next week when we get to hear you give your talk. So if you haven't signed up yet and you're still on here, sign up for Chris's talk next week.
Thanks, Ashley. All right, I guess we'll wrap it up. Thanks.